Hello, boys and girls. This is Mr. So again. Welcome back for another lesson of biology. Today we are going through lesson 13 of chapter 26 about the dihybrid inheritance. Before you start watching the video, remember to like and subscribe my channel and also share your video to your classmate and remind them to do their revision. Let's start. Previously, we learned about the monohybrid inheritance. We talked about how a single character, such as the height of the plant, change in different generations. And today, we are focusing on dihybrid inheritance. That means we are not only talking about one character, but instead, we talk about two pair of contrasting character, which involves two different genes in different chromosomes. Mendel, he focused on the shape and color of the seed of the pea plants. He first chose two homozygotes for the crossing. He chose a round and yellow seed homozygote purebred crossing with a wrinkled green homozygous purebred. And all the F1 shown here, they should be heterozygote. And they all look round with yellow in color seed. And afterward, Mendel used all the F1 heterozygous, the round and yellow seed, allowed them to grow and self-pollinate afterwards. And what he gets is for the F2 second generation, he got four different combinations. He got the round and yellow, the round and green, he got wrinkled and yellow, and also wrinkled and green, all different four types of combinations of seeds. Mendel, he is very clever and he is also a mathematician, 數學家嚟㗎. So therefore, he immediately can think of that these two pairs of allele, they must be occurring on different chromosomes, while for the wrong character, it must be a dominant allele. While for the wrinkled character, it must be a recessive allele. And then for the allele of yellow color, it must be a dominant allele. While for the allele for green color seed, it must be a recessive allele. For the round and yellow seed on the left-hand side, it must be a homozygote dominant. While for the seed on the right-hand side, the green and wrinkled seed, it must be a homozygote recessive. For the round and yellow seed parent, it is going to give one allele of round to the gamete and one allele of yellow to the gamete. While for the wrinkled and green parent, it is going to give one allele of wrinkle to the gamete and one allele of green to the gamete. And afterwards, after they fertilize to give the F1 generation, you are going to get a big R, small r, big Y, small y combination as a heterozygous for both character. The detailed mechanisms work like this. Let's take a look at the left-hand side with the round and yellow seed parent as the big R, big R, big Y, big Y. Before they produce the gamete, their parental cell looks like this. And during the meiotic cell division, they will have the chromosomes separate to the left and right into two cells. And at the very end, what would you see in the gamete? It should be, you will be only able to see one big R round allele on one chromosome, while another small chromosome with a big Y yellow allele. And then let's go to the right-hand side here with the wrinkled and green seed parent. It has both small r and small y in the parental cell. And after meiotic cell division, when they separate the chromosome into the gametes, you will be able to find in the bigger chromosome, you have only a small r wrinkled allele. And then on the smaller chromosome, it has one small y allele for the green color. That's why for both gametes, you will have on the round and yellow parent, you give the big R, big Y allele in gamete, while for the wrinkled and green parent, it gives a small r, small y allele in the gamete. And therefore, in F1 generation, it is heterozygote. Big R, big Y, small y. For the capital letter, it must be a dominant allele, showing that the wrong allele and the yellow allele, they are both the dominant character, showing the round and yellow seed as the heterozygotes. And then Mendel, he tries to cross the F1 with another F1, that means the same plant, by self-pollination. And the gamete produced from F1 is done by assorting the allele big R, small r, and big Y, small y independently. Let's take a look at the next slide to know the exact detail of how the gamete is formed. In the following case, you see you have the round and yellow seed parent as the F1, with the big R, small r, and the big Y, small y. And during the meiotic cell division, the big R, small r, they will be put on the homologous chromosome, pairing up together, while for the big Y and small y, they will also be located on the homologous chromosome, pairing up left and right. And when they do the meiotic cell division, they will separate into two different cells. So case number 1, it can be forming a gamete with big R and big Y. This is number 1 case. And then for case number 2, in the separation, you can have another type of gamete showing small r and small y. This is case number 2. However, you can actually have another possibility during meiotic cell division. For the homologous chromosome, they can arrange themselves by changing their lining up or pairing up position. Take a look at here. Right now, the homologous chromosome, big Y and small y, they switches their position. Can you see? And after they switches their position, they can have another combination here. Alright, so after the meiotic cell division, after the separation, another possibility of gamete formed will be based on the separation. You have on the bigger chromosome a big R, while on the smaller chromosome you have a small y. And then on the fourth possible gamete formed, you will have small r on the bigger chromosome and big Y on the smaller chromosome. Now, overall, how many types of gametes have you formed? You have formed 4 types of gametes. And we call these due to the different arrangement and pairing up of the homologous chromosome. You can give 4 different types of gametes. We call this situation independent assortment of chromosome. For all the F1 with a genotype big R, small r, and big Y, small y, they can give the gamete with an easy understanding of independent assortment. Let me draw it out for you. For the big R and big Y, it can join like this, forming type 1 of the gamete. And then it can form another gamete like this, big R, small y. And then third type of gamete, small r, big Y. Last type of gamete, small r, small y. So overall, you now have 4 types of gametes. Because the allele, they are located on different chromosomes, and when they separate, they are independent of one another. So we call this the law of independent assortment. This slide in here summarizes what I have been just told you and taught you. 4 types of gametes has been formed. On the top, you have the gamete from an F1 crossing with the gamete from another F1. After the random fertilization, you will have 16 different combinations of gamete, and you have 16 different genotypes. Although you have 16 different genotypes, but overall you only shows 4 types of phenotypes. So you found round and yellow, wrinkled and yellow, round and green, wrinkled and green, but you will see the amount of them is different, and they are in a ratio of 9:3:3:1. You can count it on your own. It is a very special ratio. After listening to the principle of dihybrid inheritance, I think you can take a break. And then in the coming next video, I will go through the classwork 26.4 with all of you. Please stay tuned and go to part 2 of dihybrid inheritance. See you. Bye-bye.